guys, today I'm going to talk about transgenic approaches for crop production under the guidance of Dr. Pushpa ma'am. Contents are pathogen derived resistance, the subtopics under that core protein mediated resistance, replicase mediated resistance, movement protein mediated resistance, RNA mediated resistance, pathogen derived resistance against bacteria and fungal diseases, antibodies over expressing defense genes under the control of inducible promoters, the use of clonal-resistant genes, engineering broad-spectrum resistance based on antagonistic microbes, expression of vaccines in the plants, and concluding remarks. The first one will be the pathogen-derived resistance, the subtopic under that core protein-mediated resistance. In this, the concept of using mild strains of the virus that cross-protect plants against more severe strains was first demonstrated in 1929 using tobacco mosaic virus. For many years, strains of the tobacco mosaic virus were used to protect glass houses grown tomatoes in the UK against more virulent strains of the virus. Mild strains of citrus virus have similarly been used to protect citrus trees in the Brazil, and the apple mosaic virus have been used in the New Zealand in the same way. In 1986, this was demonstrated when the core protein gene of tobacco mosaic virus was expressed in the transgenic tobacco plants under the control of cauliflower mosaic virus 35 as promoter. The result delayed symptoms development in all the transgenic plants, and 10 to 60% of the plants failed to develop any viral symptoms. In the form of resistance now refers to as pathogen-derived resistance or has subsequently been demonstrated to occur against a wide range of viruses and used to recognize as effective way of controlling specific viral disease in the plant. The viral food protein produced in the planta was the key component of the resistance. The expression of this protein was believed to block the normal uncoating and the replication of invading viruses, and the evidence to support this was provided by use of tobacco mosaic virus and the pseudoviruses. In GUS reporter, gene attached to the viral origin of assembly was packed into the intact viral particle. Although the core protein mediated resistance works effectively against closely related strains of the virus, in some cases requiring as little as 60% of core protein homology in ineffective against unrelated viruses, and this was before not type of a broad spectrum resistant sought by the commercial interest. It needed when the commercial strains of potato possessing either PVX or PVY core protein mediated resistance were produced in the 1980s that showed effective resistance. Products made in the commercialization of unfavorable against the unlimited added to the value of the grower. Core protein mediated resistance refer to the resistance of the transgenic plants that is produced core protein to the virus in which core protein gene is derived may rises due to the interaction between the transgene core protein and the virus of core protein. The second one will be the binding of transgenic core protein to the host factors required for disassembly for the virus. The interaction of the core protein with the nuclear conclusion protein B, it is a specific photoviruses. In this, the left side we are going to see about the resistance against tobacco mosaic virus in transgenic tobacco expressing tobacco mosaic virus cross protein gene. Food protein mediated resistance, that is crop production. These transgenic plants expressing food protein is showed across cross production to infection by another closely related viruses. This is known as cross production. In this right side we are going to see the related species, which will be the first one. The transgenic viral cross production of RNA, then it will came to the translation. It will be cross production with the uncoating and replication and translation, and that results in the movement of the transgenic plant. The next subtopic is replicase-mediated resistance. The replicase-mediated resistance was first demonstrated against TMV virus, tobacco mosaic virus, where the viral replicase was expressed in the transgenic tobacco under the control of 35S promoter. Replicase resistance has been subsequently been shown in the effective against a wide range of single-stranded RNA viruses and some genuine viruses, and in many cases, deleted versions of replicase genes are sufficient for production. The resistance is generally more effective than uncoated protein mediated resistance against high inoculum doses. In one case, broad spectrum resistance against wide range of viruses was demonstrated when tobacco mosaic virus replicase gene containing a bacterial suction element in which it was used. Replicase-mediated resistance probably works through the post-transcriptional gene slicing mechanism rather than through the interface by the replicase protein. There are many number of incidences where the resistance have been broken by a small number of the points mutations within the infecting viral genome. The next subtopic is movement protein mediated resistance. The importance of movement proteins for the cell to cell movement of virus. It's been proposed that blocking a plasmata and the viral movement may be a more effective means of preventing the spread of both related and unrelated virus that results in broad spectrum resistance. Tobacco plants expressing Bumi mosaic virus were found in the resistance against unrelated tobacco mosaic viruses, and the plants transgenic for the defective mutant of the TMV movement protein were resistant to a wide range of unrelated viruses. Attempts to produce the transgenic plants with the further movement protein mutations based on the enhanced understanding to how these proteins work are currently underway. In this attempt to produce more effective broad spectrum resistance to the virus, attempts that have been made to another region for the viral genomes to make pathogen-derived resistance plants such as protease. Hence, the approaches have made this success because they are unlikely to provide the broad spectrum resistance that movement proteins might provide. They have been less executively studied. The next subtopic is RNA-mediated resistance. The possibilities that replicase-mediated resistance may be conferred by the post-transcriptional gene slicing mechanism has led to the development of RNA-mediated means of protecting plants against viruses. This mechanism is supposed by the experiments in which transgenic plants with enhanced resistance have been generated by expressing satellite RNAs and the defective interfering RNAs associated with certain viruses. However, this approach is limited to use against those few viruses that possess the satellites viruses, and the specificity of the sequence required a very narrow host range resistance. In addition, many viruses appear to possess a mechanism that is suppressing PTGS. Finally, double-stranded RNA have been potent to induce the PTGS and the more plant viruses replicate through the double-stranded RNA intermediates. Secondly, in the rabidovirus mutants in this PTGS is impaired where the hypersensitivity to the viral infection. Thirdly, some viruses have been shown to possess the mechanisms to suppress the PTGS. Fourthly, PTGS was often stored in all the cells that is recovered from some viral infections. The ribosomes, which are self-clearing RNAs based on a certain satellite RNAs, and the virids also have been tested for the effectiveness in the control of viral diseases. The aim has been to design the ribosomes where expressed in transgenic plants could bind to the viral RNA and clear it that will be inactive. Similarly, antisense RNA approaches have been tested against some virus. However, there is no results that suggested these mechanisms have significant advantages For the better tested methods of cold protein replicase or movement protein mediated resistance. And the last subtopic under pathogen-derived resistance is pathogen-derived resistance against bacterial and fungal diseases. That attempts have been made us to use pathogen-derived resistance to control the bacterial and fungal diseases. Isolates of Syringo and the Facilocora that produces facilitoxins encode a ROTAs resistant to the OCTs and the enzymes to deal with the adverse effects of the toxin. Toxins have a particular important weapon named as the P-cyrongopathenophores, where the these are generally non-host specific genes are not essential for the pathogenicity, where induces the chlorosis in the plants and increases 1817 of the viruses were the best studied the bacterial taxons of pathogenesis B. Tobacco, tabotoxin, facilitoxin, syrengomycin, Cornotin is produced by the number of serengopathophores such as the glycosin and tomatoes which affect the soya beans and the tomato species respectively. Tobacco toxin was synthesized by the wide range of peas syringopathophores such as the wildfire of tobaccos and the hollow bright disease of oats and the lactate. These are the generated through the amino peptidase cleavage is the active part. These are the chemical structures of bacterial non-specific toxin that is formed in the corinotin by P-syringate and the tabotoxin and facilitoxin and the syngomycin. The ROTAs have been engineered into the tobacco targeted to the chloroplast where OTGs is produced by fusing to the ribose biphosphate carbylase small subunit transgenic phosphate and the resultant plants were resistant to the toxin. Attempts now being made to engineering such a system into the being the natural host of this bacterium. Some stains roughly 1% of the coming ones of the mice murd diseases possesses double-stranded DNA microviruses that confer the killer phenotype where double-stranded RNAs encoded the killer proteins and the immunity to the taxon. These killer proteins are effective against the isolates of the small diseases where double-stranded DNA conferring immunity including the another species. The killer protein genes have recently engineered into the Swiss wheat converters susceptible to the shrinking smolts, where we are studying about the mice small diseases, which resulted in the plants and increased the resistance to the fungus. However, were such a plants to commercially useful for the unclear, since one consequence of these use is like to build up the several population of isolate that possess the immunity of taxons. Additionally, these killer proteins appears to protein blockers of calcium channels where have been structurally similarities to the scorpion toxins which may use in the edible plants and undesirable. And the next topic is antibodies. The another approach to create plants resistant to the pathogens might be express antibodies that recognizes the pathogen in plants in such way they mop up and utilizes the infecting pathogen before it causes disease. The success of serological techniques they have ability to recognize antigenic sites that are specific to the pathogen is to be detected. Polyclonal anti-serum that have been routinely used for the number of pathogens, particularly virus, because they relative the easy in which that is produced. Essentially, the pathogen or a part of a pathogen, such as viral protein or a particular peptide, has to be purified or chemically synthesized. The purity of antigen is crucial since the antibodies will be raised to any proteins that is injected into the animal. This results in the problems with the antibody detecting in the contaminants subsequently analysis. The purified pathogen or peptide is injected into the test animals such as rabbit, and the following subsequent booster injections the animal is blind. The resultant antisera, which contains the population of antibody that may react with the different contaminants of the immunoglobulin and which have been utilized in the detection techniques. The monoclonal antibodies are superior to the tropes where they can provide constant supply of specific antibodies. They are, however, relatively expensive to produce and maintain because they have specialized cells, cultures, and low temperature storage facilities that are required. The production method is shown in the figure. Monoclonal antibodies have been produced and they are commercially available against wide range of viruses such as common seed-borne and the potato virus, where diagnosis is important to prevent the dissemination of the virus in seeds and tubers. They have been produced against specific oligosaccharide side chains of the erwin bacteria and against proteins from some other bacteria and protoplasms, as well as specific proteins from plant pathogen fungi, allowing for the example and identification of proteins. That is grape juice that we are going to drink about the grape juice, where we have used the monoclonal antibodies and discrimination between the high spot and another stem based diseases of the wheat that give the similar symptoms. Here come the monoclonal antibody production. Here the mouse nation stimulates the antibody production. Here the antibody cells isolated from the spleen and tumor cells are grown in the culture are fused to form hybridomas. Screening for the antibody production and clonal antibody producing hybridomas, and the monoclonal antibodies are isolated from the cultivation. The use of page display has proven to ability to clone the genes encoding specific antibodies or planning phage displays libraries with the specific pathogen antigens such as pathogenicity factors that can be used to identify antibodies that recognizes the antigen and relevant clone that can be isolated from the library. The targeting clonal antibodies to the particular compartments in the plants can be accomplished by the adding targeting sequence to the transgenes. For example, leader sequence can be added to the target proteins for the secretion to the apoplast or KDLE sequences. 224 molecular plant pathology at carboxyl terminal can be used to retention of the proteins in the endoplasmic reticulum, which happen being used to result in the higher yield cytoplasmic expression of a recombinant antibody for the viral inhibition in the plants has however been problematic. Antibodies, particularly whose are obtained from the conventional means that is planning for the page display, are less stable in the core cytosol that is other than the cellular compartments. For example, tobacco mosaic virus specific methods to integrate antibodies into the plasma membranes, for example, incorporating transmembrane domains such as antibody phases apoplast resulted in the transgenic plants that resulted in the tobacco mosaic virus. Antibodies that have been developed for the against bacteria, particularly phytoplasm. These organisms are phloem restricted, so the plant bodies have been targeted to the secretory system. Antibodies to the major membrane proteins for phytoplasm were expressed such as secretory signals in the transgenic stomata are confined resistance to the spillover, where use of the antibodies to target the fungal diseases is less well developed. The third one will be the overexpressing defense genes. Many of the pathogenesis related proteins which are induced by the plant disease resistance processes have direct antimicrobial effects. For example, gluconesis, protease inhibitors, and hydrogen peroxidase generating enzymes are introduced as a part of hypersensitivity responses as well as the enzymes involved in the biosynthesis of phytoxin. The numerous attempts have been made to introduce genes encoding these into the transgenic plants under the control of constitutive promoters to increases the level of proteins and enhances the resistance. In 1991, there have been gene introduced into the tobacco under the control of cauliflower mosaic virus 35S promoter, resulted in transgenic plants in which enhanced the resistance in the rhizoctolanaceae, although there is no significant resistance to another pathogens such as Silofera Nicotina, where another containing non-humids species. In addition of glucines, transgenes is combination were subsequently shown to produce the resistance. Similarly, expressing lysosomes in the potato increased resistance to the bacterium, and PR1 tobacco increased the resistance to the oomycetes, that is blue module, and the black shing diseases. An alternative approach to produce photoalescence in the plant, the grapevine stillborn synthesis gene, have been introduced into the tobacco to confer the resistance against some viruses. This gene is involved in the biosynthesis of photoalescence in the grapevine and some other plant species, but absent in the tobacco, although the chemical precursors that will be produced in the tobacco. In transgenic plants, they converted precursors into the stillborn, resulting in the enhanced resistance. In this tragedy, they have also been used to alfalfa for the resistance of the phloema species, and to approach the engineering plants to produce different photoalescence may be applicable to diseases as well as being tested to more biosynthesis pathways. And other approach is used especially against a necrophytic fungus, that is some fungal species that make use of the fact in this fungus synthesis a very large quantity of oxalic acid when infecting plant. This oxalic acid is an essential cofactor required for the pathogen to degrade the plant cell walls to colonize the plant and defective mutants and the non-pathogen. Overexpression of plants derived oxalic acids or oxalic carbolysis in the transgenic plants such as soybean, oilseed rape, and the sunflower have been shown in the result in the rapid breakdown of oxalic acid from invading pathogen and resulted in the increasing resistance to the pathogen. Under the overexpressing genes, the general model for enhancing disease resistance in the transgenic plants. The first one, here the leaves are infected with the fungus where it causes a diseases, and the non-transgenic plants at low level of expression due to the innate genus expression. For the 35S promoter, the introduction of the cytokine were under the control of 30S promoter for the transformation. The second one will be the expressed the gene that will be introduced in the high level of confer the resistance to the certain pathogens. The fourth topic under the seminar is expressing defense genes under the control of inducible promoter. It's one of the fundamental problems with the overexpressing defense genes in the plants is that the plant resources that might otherwise used by the increasing yield production have been derived to maintain this high level of resistance. The constant production of defense genes produces that as photoalescence where the pathogen is present or not, that might be undesirable chemicals are present at the higher level than normal, making the produce unsuitable for the consumption. In this, the alternative approach to engineer defense genes are genes that is mimic to the defense response under the control of pathogen-derived promoters so that they only expressed on the pathogen invasion. For example, on this is a ribosome inactivating proteins such as provid and antiviral protein. This is the particular protein have been used in the human medicine as an antiviral agent, for example, against AIDS for a number of years. More than recently, it has been transgenic approach for the crop production under 247 engineered into the tobacco, where it is been shown to protect against the diverse range of the virus such as PVX and PVY and cauliflower mosaic virus. In more elegant advances, the second ribosome inactivating protein have been introduced into the tobacco under the control of Gemini promoter in the same, whereas antiviral responses is the only induced in the induced in the cells and then attacked by the pathogens, such as there is no D-anthem expressed in the uninfected plants or the cells away from the site of pathogen attack. A similarly strategy have been collated and tomato against Photothera infantis. In this case, the promoter from the potato, the gene PRP121, in which mediates the expression in the response to the bright attack, have been linked to the bacillus bacterial ribonucleases gene. The expression of this gene in the plants resulted in the formation of nucleotic vision occurs in many ways to innate the plant hypersensitivity responses, because this gene was found to express at a low level constituent to have the deleterious effect on the plants in the second bacillus gene. This inhibitor of the Venasa have been instantaneously introduced into the transgenic plant. The resultant transgenic plants have been induced level of the fungal sporation where the inoculated pursued, because the infected pathogen derived induced the production of runs in the infecting cells to mimic the hypersensitivity responses. Hypersensitivity responses and restrict the development of the pathogen. The fifth one will be the use of clonal resistant genes. The cloning of functional resistance genes has resulted in the capacity to transfer such as genes directly into the commercial crops without the needed for executive back-crossing programs. For example, the XLA21 gene from the gene to resistance of Exothermus horaeus have been transferred at the Research Institution in Philippines from the species horaeus into the elite cultivators for rice for the commercial use, and the process that have been difficult to achieve by the conventional breeding process. In this addition, the engine have been transferred from the tobacco into the tomato, where they confer the resistance to the tobacco mosaic virus. However, when the BS2 gene have pepper resistance to the xanthomonas, where the cloned into the species 248 molecular plant pathology outside the Solanaceae. In the ineffective indicating that many R genes have been restricted taxonomic functionality and not operated unrelated plant species. Such approach is clearly flexible for many, save a great deal to time in introducing genes into the good aromatic cultivators and the approaches that could be combined with the identification of the novel resistance specifications. Alternatively, these expressions have been undertaken with the domains of resistant genes, and the resistant gene analogues have been randomly combined to in vitro to attempt to generate the disease resistant genes in the novel specificities. The technologies are being developed to the rapid screen of products. These recombinations of these effectiveness as the resistant genes, such as any potential use that could be engineered into the transgenic plants. And the sixth one will be the engineering broad spectrum resistance. Introduce the single clonal resistant genes into the transgenic plants does not net ground problems resistant identified in the conventional breeding programs of the rapid pathogen evolution to overcome the resistance genes. It is important to practice the primatizing resistance genes in a particular format that is likely to the effect is continued. Unless this is done in the rotational, the tragedy way to mergely developing new resistance genes and they become the available to continue to the fuel revolutionary harms race between the crops and their pathogens. In the additional priority that must be identified more spectrum and inducible resistance that will be introduced into the plant. The Xanthomonas oryzae 21 gene, that is XLA1 gene, for example, it is capable for recognizing a virulence protein secreted by most, if not all, pathovars of the Xanthomonas oryzae that is effectively provides the desirable genes. The ESP2 protein produced by the Cladosporium species is also secreted by all pathogenic races for this fungus. So, targeting this protein by identifying and cloning CFELP genes have been increased the potential to develop desirable genes against the fungus in tomato. The Bailey MLO gene is a durable and attempt being used to identify homologous in this another plants in another great genes that make a particular species into the another host will be identified. These could be cloned into the current plants to tender them into the resistant. An additional transgenic approach to development broad spectrum resistance based on the resistant genes have been genetically engineered acquired resistance system. This involves in the incorporation into the plants for the fungal virulence genes in the combination with the responding resistant genes inactivated by the insection of mutagenesis. For example, tomato plants have been engineered that the contain tomato CF9 resistance gene inactivated by the Transgenic approach for crop production 249 transposable element in the combination with the Cladosporium species ARV9 gene. Because the resistant gene is inactivated by the transposon, these plants grow normally. However, at the low frequency, the transpon exercises the somatic cells and these cells are functions of the resistant genes is destroyed and the product of this gene interacts with the product of a virulence gene in the resultant in the localized hypersensitivity responses in and around those cells which are response on the exit. This resultant is a very small nucleotic regions and introduction of some systematic resistance response throughout the plant in such way they become the immune to the infection and the cladosporium species. The late blight and the powdery mildew diseases. These resistant virulence genes have been shown work on when the transferred together into the potato plants and confer the enhanced resistance against potato blight diseases. F9 and AVR9 have been engineered into the soil weed repair the unethical RHR. They have been introduced confer the enhanced resistance. Such system could be engineered into the another plant system in which the resistance and corresponding virulence genes as identified. Furthermore, controlling the expression of these resistance gene and virulence gene throughout the use of pathogen-derived promoter could ensure the systematic resistance is only activated when the plants are infected by the pathogen. This is the principle behind the genetically engineered acquired host spectrum resistance. A further approach to produce the broad host range resistance have been to make use of genes that is the part of the single transition mechanism in local and systematic resistance pathways. NRF is the key gene is regulating these pathways and overexpression of RPN1 albedo species and enhances the resistance to the broad variety of pathogens such as Pseudomonas and the other species with no abuse. In rice expression of GPR1 homologous resulted in the increased resistance to the Xanthomonas and other species. However, the expression of NPR1 homologous in wheat had been a positive effect and expressing the expression of pathogenesis related genes and enhancing the resistance susceptibility. The another gene in signaling pathways have also belonged to examining the potential candidates for conferring the broad range of the resistance such as R1, RAR1 and SGRT1 and PAD4. For example, in inactivation of PAD4 in the Aridopaces resulted in the extreme susceptibility to range of the fungal mycets and the fungal pathogens, so it can responsible that overexpression has been opposite effect and resulted in the enhanced. The seventh one under this is resistance based on antagonistic microbes. Antagonism between the microbes is the common feature for the natural ecosystem. They compete for the nutrients and the habits. Many are capable for producing toxin, antibiotics, and another antimicrobial capable for killing other over the microbes to enhance their own chances for occupying a particular niche. In this, they exploited with the development of biological control microbes that can be used especially in the rhizopods and the target and combat plant pathogens. Isolates from the soil, bone, fungus, trichoderma and the bacteria such as bacillus species and the pseudomonas fluorescens have been developed into the commercial biological control agents against the range of seedling diseases. These organisms are particularly useful when they be produced relatively easily and formatted into the products widely with a relatively long shelf life that gives the reliable biological control efficiency. The development of many other potential biological control agents that have been shown to work. Laboratory experiments are targeted against the follicular pathogens have been less profitable because the inner problems with the storage and appropriate means for application to give the food reliable control. Without these properties, growers will not be willing to use them. They replace the conventional control methods. However, these are some examples of genetically modified microbes that is already being used as a biocontrol agent, and one good example for the biocontrol have been in the crazy gill diseases, the agrobacterium tuberculosis, the non-pathogenic soil inhibiting related bacterium Aerodobacter were identified the produces an antibiotic for many years. The strain of this bacterium K84 have been used to biocontrol agents against certain strains of agrobacterium species. Seedling of the plants will be dipped into the suspension of the bacteria priority to the planting and strain K84 K84 could be the colonizes the soil around the roots and protect them against ground gill. The capacity to produce this encoded on the large plasmids as that the immunity region that have been protected K84 from the effects of agrocin. However, this plasmid also possesses the transfer region. In the laboratory experiments, at least it has shown that the transfer region able to producing the plasmid to transferred into the agrobacterium species should occur in the field that resulted that cone in the gill both when the immature of the agrocin. As a result, a new strain of the radiobacter have been engineered in 1980s in which the transferred religion have been detected. This new strain K1026 have been sold commercially as a biocontrol agents in the Australia and more recently have been registered for the parts of the USA with no obvious detrimental effects on the rhizosphere. The development of strain K1026 of Agrobacterium radiobacter for biological control of the Agrobacterium species. The expression of vaccines in the plants. Plants refers to the cheap and reliable way to producing the medicinal proteins that might prevent human diseases in high yielding and easy profitable form. Approaches have been developed using the plants to produce vaccine. This tragedy have been used to engineer epitopes from the animal viruses, for example, foot and mouth viruses, at a specific position into the code protein genes of the plant virus, such as cowpea mosaic virus. The resultant hybrid virus have been foot and mouth epitopes on the outer surface, and its code. This virus can be grown in the plant and purified and used as a vaccine. Similarly, epitope on the outer membrane proteins of Pseudomonas species have been engineered into the TMV code protein. The engineering plant virus for the use of vaccines, that is cDNAs encoding epitopes, are engineered into the plant virus code protein genes so that can explode on the outer surface. This virus is then prolongated in the plants and purified and used as the vaccinate animals. Concluding remarks. We have been dealt advances in the understanding of the molecular genetics of the plants and their pathogens. We have examined how pathogen causes diseases and how plants resist diseases and how it might be polluted through the supporting conventional means, the disease control, or through the biotechnological approaches. Whether the use of genetically modified plants will gain the public acceptance worldwide in the detail. In not certain, these approaches will significantly alter the continued arms race between the plants and pathogens. In the additional session where the pressure existed on the pathogen by more elaborate to control the methods in some of them, which have been resulted to the development of super pathogen in which they let more elaborate control have been used. Equally, not conventional policies are the problem. From these resistance heals have been fungi produce the mycotoxins. If they are not controlled, they produce the potential more harmful to the health diseases may contain the fungicide residues. There is no doubt that the molecular biology have been placed the development of crop production method. It is only the advancing our understanding of the ways in which the plants and microbes interact through the desired signaling pathways and changes the gene expression in greater deals. Whether they throw the marker and breeding improve the molecular diagnosis or another methods, and research and enhanced understanding will result in the development methods that can be applied in the future. New problems will continue arises. Because new diseases will alone spread the region where they can be absent, or now techniques have been studying for the pathogen will be development for the techniques and technologies in advance. The plant pathogenists who have understand and keep the modern concept about the nature of the plant diseases. This way in which they can be controlled will be the viral for the agricultural and horticulture and forestry industries. And the references is Molecular Plant Pathology by Dickinson, School of Bioscience, the University in the UK. The fifth edition of the Plant Pathology by the Agrios Department of Pathology in the University of Florida, and some YouTube